Welcome back to Getting Started with CellEngine. In this video, we'll be continuing our discussion of illustrations by looking at statistics plots like bar graphs and heat maps. Last time, we discussed making flow plots, both singly and in automatically generated grids. That video covers a lot of details of the illustration page that are useful for making statistics plots, as well as plots from single FCS files. So you may want to watch that first. I'll mention a couple of places where those features could be especially useful and link the video in the description below. This video will cover the kinds of statistics plots and charts that you can make in CellEngine. As we go through them, we'll discuss how to use metadata to organize your plots, adjust the order of populations and other groups, customize and assign the colors in a plot, and scale data. Let's go over to CellEngine. We're going to start not on the illustration page, but in annotations. We've talked at various points about how CellEngine is metadata-driven, but statistics charts are one of the places they're most powerful. So if you've been following along and haven't added metadata to your samples yet, you might want to go ahead and do that now. The experiment we'll be using for most of our examples today is a time course, with samples collected daily from donors infected with influenza. The annotation page has added details about the sample, like the day of collection and the donor ID, as well as some details about the illness outcome, like whether donors developed fever, shed virus, or displayed other symptoms. I've also added information about the batch that each sample was run in. We'll use this information in making our plots in a variety of ways. Let's start by discussing box plots and bar graphs. Let's make a new illustration page. Those icons are here to the right of the pivot table icon in the top bar. I'm going to put down one of each type. Just like pivot tables and flow plots, when you set down the plot, you'll find all the options for your selected figure on the right-hand side. As a reminder, when you select multiple plots that have the same options, you can change them simultaneously. Box plots and bar graphs share a lot of similarities, so let's select both of these, and then we can assign the options together for easier comparison. For most statistics plots, setting up dimensions will involve choosing an axis label and a legend entry group. Axis labels determine how the data will be separated out on the axis, either X or Y, depending on the orientation of the plot. You can choose populations, channels, individual FCS files, or any metadata that you've added. Let's start by comparing a few different major cell populations at a couple of time points to see if there are big differences. I'm going to set the axis labels to populations and then select a few values. You can see how the axis will be sectioned off for those already on the plots. Now we'll need to choose legend entries. As the name suggests, these will be entries that appear in the legend and will be colored by value. Since we're comparing time points, I'll choose the annotation day for the legend entry name, and then choose the values that I want to display. I'll also go down to the general section of the options and choose the statistic. This drop-down box displays the various options available for us. For this example, I'll choose % of to look at the relative frequency of the populations that we've selected. When choosing a population for % of, parent will always be the top option in the gating hierarchy for this display, so we can have % of whatever population is being displayed. We could, of course, also choose singlets or any other population that we want to see the % of. Axis labels and legend entries can be swapped under dimensions by clicking on these arrows if you'd like to see the data grouped differently. Notice that when we swap these, CellEngine has recognized that the values are now numbers and spaced the data out on the axis based on the values. To treat the numbers as a category, just like it would text, check Categorical underneath axis labels. Both bar graphs and box plots can be moused over to get a little bit more information about the data. The bar graph will give you the specific statistic and error, while the box plot will give you information about the specific data point that you're mousing over. This can be useful for identifying outliers or just points of interest in the spread. The plot settings section also contains settings for the color palette, which we'll discuss in detail shortly, and orientation. Axes and legend contains the ability to adjust the size or presence of the legend and labels. When you have multiple types of plots selected, you'll only see the options that are shared between all of the plots. For example, the error bars on bar graphs can be customized, but because there's no corresponding option on box plots, that won't be displayed as long as both are selected. So when I select just the bar graph, We can now set the error bars as standard deviation or standard error of the mean, or turn it off entirely. Notice that the axis label of the plot will reflect that choice as I change between those options as well. We set these examples up using metadata for one of the dimensions, but metadata can also be used for both. For our box plot, let's change the plot to look at donors that develop different symptoms. I'm going to set the axis labels to peak temperature and then legend entries to other symptoms. Since we don't have populations as a dimension any longer, we'll need to specify which population we want for the plot, which is under General Settings. I'm going to choose plasma cells for this example. Now we can see the percent of plasma cells separated out by donors that developed fever and/or other symptoms. One other thing to point out, which is at any time if you want to hide a legend entry, you can click on that group in the legend. That can make it easier to check specific comparisons or try out various combinations before deciding on a final layout. Currently, our plot includes all the samples in the experiment that match our dimension criteria, which means that all time points are currently included. Just like pivot tables, we can add an additional filter to adjust our selection of data. Under Filter Annotations, click on Add Filter and select the annotation name and value to filter on. For example, to look at a single time point, we could use day and just look at the data from day 5. Previously, we discussed batching, which will create figures automatically based on selected variables. Statistics plots can also be batched. For example, we could create a version of this page for each day in the experiment or each population or other values. We went over this in detail in the previous video when talking about flow plots and pivot tables, so if that's of interest, you may want to watch that video. While we're looking at organizing figures by metadata, let's discuss sort order. CellEngine orders groups depending on the values. By default, CellEngine will sort numbers based on value, taking units into account if those have been included. Dates will sort chronologically, and most text will get sorted alphabetically, although plate well IDs will sort row-wise from A1 to the end of the plate. This can be modified by changing the sort order, which we can access by clicking on these 3 horizontal lines in the top bar, or the same symbol next to the dimension of the figure. That will open this dialog box where we can make adjustments to the sort order of the particular group. Sort order can be applied at several different levels. You can change the order for the entire experiment, for the current illustration page, or just the plot that you're looking at. Select the values from the list that you want to move, and use the buttons to the right to modify the order. I'm going to switch the property to populations, so we have a few more options. You can select one or more values in the list by Shift or Control clicking, and then use the buttons to the side to adjust. So if we want the T cells to always appear first in the sort order throughout this illustration page, we'd set the level to illustration, select the populations, and then click Move to Top. Once the changes are saved, you'll see that reordered the bar graph that we created earlier. Colors for plots are determined by the color palette options, which are found in the plot settings for plots that have discrete values. CellEngine has several built-in options which can be selected from the dropdown, but these can be customized as well. Click on Edit next to the color palette to make changes to the selected palette. This dialog will show you the hex codes and a swatch for each entry in the palette. You can copy and paste specific values into these boxes. Here I have a spreadsheet with a few hex values, and I can copy the whole column and then paste to change the first colors in the current palette. You can also click on Copy Palette down at the bottom of the list to copy all hex codes from your existing palette, so you could use the palette for making figures in other software or other projects, or copy to an already existing experiment. If you don't have a specific hex value for color in mind, or you want to make small adjustments, you can also manually change colors by clicking on the swatch and use the color picker tool to make adjustments. By default, colors will be assigned to groups in order from top to bottom. If there are more groups than colors, the order will just be repeated. The order can be adjusted by clicking on the up-down arrows next to each value or by changing the hex values. Colors can also be assigned to specific groups of data. So for instance, we could make asymptomatic gray and symptomatic red, regardless of the palette order. Like the sort order, color palette edits may be applied to several levels within an experiment. I could make these assignments for the entire experiment, for the current illustration, or just for the particular plot being worked on. The same color can be assigned to multiple values. For example, if we change the property to populations, we could assign the same entry to class-switching memory B cells as well as basophils. I'll set this level to be experiment-wide, so now whenever basophils appear elsewhere in figures using this palette, they'll use this color. When you're done making edits, save the palette. The last group of shared options includes the scale and range for the plot. This can be changed in any of the statistics plots, so let's set up a line graph and discuss a few line graph specific options along the way. As we move right in the top bar, you'll find the line graph option right next to the bar graph and box plots, so I'm going to set one down. Let's set the axis labels up to show days for the first week of infection, and then for the legend entry, I'm going to pick samples from cohort 3. Going into general options, I'll choose another population and a statistic. We've been looking at a lot of examples with population statistics, so let's look at the median of CD38 in B cells this time. Line graph options are very similar to box plots and bar graphs, but there's one additional option you'll see underneath axis labels, which is connect gaps. You'll notice that this cohort has no files from day 2. If I check connect gaps, CellEngine will extend a line across any gaps caused by sparse data, which can be useful in some circumstances. Apart from this addition, you'll find the same options as those we discussed already for bar graphs and box plots. I'm going to change the legend entries to virus shedding and select both values so we have a better example for scaling. You'll find the options down at the bottom of our sidebar. The scaling dropdown lets you select your preferred method of scaling data. Raw options use the unmodified value. Population statistics would usually use one of these options, while the scaled options would usually be for channel statistics and utilize the channel scale. You can display the values as is or display fold change or difference. The values or ratio of values can also be displayed in log 2 or log 10. If you're curious about the mathematical calculations for any of these, they're available in the documentation for further reference. When you pick one of the scale options with a difference or a ratio, you'll be able to choose what data to normalize to. You can normalize to an entire data series like an unstimulated control or samples from donors that didn't shed virus. We could also normalize to the left or rightmost axis group, which for this plot would be either day 1 or day 7. The chart minimum and maximum will find the highest or lowest point on the chart and normalize to that instead. In addition to setting scales, you can also set the threshold for displaying values as decimals or as scientific notation. If we pick a value in the middle of the range for the minimum, you can see anything under that is shown in scientific notation, while anything over that is set to a decimal. This is a little bit odd to use on this particular example since the values are quite low, but it can be useful if you have statistics across a large range. CellEngine will try to automatically select the appropriate minimum and maximum values for the range, but you can also set those manually. For example, we might want to set the maximum slightly higher for aesthetic reasons on this plot. We have a couple more plot types to discuss, along with a few options that are specific to those plot types. We'll need a different experiment for the dose response curves, so let's discuss heat maps before leaving this experiment. We can create a heat map by clicking on the last icon on the top bar on the right-hand side. Heat map dimensions are usually the row and column values used to set up the table. For our example, let's use donor IDs and days as the dimensions. For the population, I'm going to use intermediate monocytes as a percentage of singlets. Because heat map data is continuous instead of discrete, you'll notice your color options in the general section are based on color scales rather than palettes like the rest of the statistics plots. We can select from the options in the dropdown. Beside that small change, you'll also find a few options that are unique to heat maps. Heat maps can be clustered by column, row, or both. I'm going to cluster the donors for this particular heat map. There are also a couple of style options to adjust the line thickness or set the amount of the heat map dedicated to the dendrogram. If you decrease the size of the dendrogram to 0%, You'll still have clustering, but the dendrogram will be hidden completely, which can be also useful in some circumstances. CellEngine includes several different approaches for clustering, including single, complete, and average linkage, as well as Ward's method. In this case, the average linkage approach shows a clustering of donors that had an increase in the intermediate monocyte population around days 4 to 7. Metadata can be displayed for rows and columns as a kind of overlay and can help enrich the data presentation. Let's add some additional information about the donors. I'll click on column annotations and select one or more annotations. In this case, let's look at viral shedding and peak temperature. If you have multiple annotations displayed, the display order can be changed with the arrows next to them. So I'm going to swap these. Interestingly, it looks like donors that had the increase in intermediate monocytes all shed virus at some point during the infection, although the presence and absence of fever doesn't seem to correlate strongly. Adding metadata to heat maps can help reveal or emphasize these kinds of connections in the data without adding additional plots. Heat maps can also be set up to show correlation. Let's make a new heat map and begin by opening the heat map correlation section. Here you can select which algorithm is run to calculate the correlation coefficient for the heat map. When we scroll back up to set our dimensions, you'll see that the dimensions have been renamed. Instead of rows and columns, we're now setting rows and columns for comparison in one dimension and values for the correlation vectors in the other dimension. One common setup for correlation heat maps would be to use channels for the rows and columns. This particular study has quite a few signaling proteins, so let's select some of those for our rows and columns. For our correlation vector, let's use populations. You'll need at least 2 of these for results, but 3 or more will give a broader range of values. So I'm going to choose several populations for the vector. Sometimes you may want to use channels as a dimension in a non-correlated heat map. For instance, if I change the correlation coefficient on this plot back to none, we can now look at the channel data for each population that we have selected. By default, CellEngine will use one scale for heat maps. It's not uncommon for some channels to be brighter than others, so if that difference is large, it can be hard to see changes in channels with a smaller range. In this example, Stat 3 is by far the brightest channel that we have selected. To have each channel in the heat map scale independently, we can go down to Scaling and Range, and then select Rescale Channels Equally. Now you'll notice that the legend will scale from 0 to 1, and each channel has been scaled with the minimum value for the row at 0 and the maximum at 1. Now we've covered heat map features, let's look at an example for dose response curves. I'm going to switch to a different experiment, and let's start by looking at the annotation page. When you have a dose response experiment, you'll want to make sure that one of the annotations you have is the dose. This can be added with or without units. To make a dose response curve, let's make a new illustration page. The dose response curve icon is right next to heat maps, so I'm just going to put one down. For the dosages dimension, choose the annotation column with your dose information. In this case, concentration. You'll need at least 4 values for the calculations, but can add as many more as you have acquired. For the legend entries, this particular experiment includes 3 different runs, so I'm going to select those separately. Under General, we'll also need to set the population of interest and the statistic, in this case, the median of a RAC4. Once you've set the plot up, you can display fit parameters, including the IC50 and R-squared value for the curve, by hovering over the legend entry. You can also download all that information into a spreadsheet by going to Export Selected Components and then choosing Download Dose Response Fit. This concludes the overview of setting up statistics plots and the illustration pages, and should give you a strong basis for setting up figures with your data. If you have any questions about figure creation or other features in CellEngine, feel free to contact us at any time by emailing support@cellengine.com. Next time, we'll wrap up the series by discussing exporting statistics for further analysis and running dimensionality reduction and clustering algorithms in CellEngine from setup through visualization.